Hi, everybody, and welcome to the Chapter 6 lecture, which is entitled Reproduction at the Cellular Level. We are going to start by taking a look at this image right here, which is an image of a cell under a microscope, which you may have guessed. But this is a very specific type of cell. It's a very special cell. This is a cell called a zygote. A zygote is the type of cell that emerges when a sperm fertilizes an egg. So this type of cell was every person's beginning who is listening to this lecture. Every single person, every single human started off as one of these guys right here. Fast forward to today, and your average human body consists of 37 trillion cells, which is 37 with 12 zeros after it. So the question that we are going to be exploring today in this lecture is, how in the world did this happen? How did every human go from being a single cell to being this walking, talking, 37 trillion celled organism that you are today? Well, what happened is that original cell, that zygote, made a copy of itself so that one cell became two. And then each of those two made copies of themselves so that two cells became four, four cells became eight, eight became 16. And as your cells continued to copy themselves, they started to take on special roles so that some became skin cells, some became blood cells, some became brain cells, etc. Until eventually something looking like a human started to take form. So this process of cells copying themselves and old cells making new cells is very important for growth and development. But that's not all it's important for, because once you become an adult, that doesn't mean that your cells stop growing and reproducing. The process of making new cells occurs continuously in all parts of our body throughout our lives. It's necessary for replacing any sort of damaged cells, such as when you skin your knee and develop a scab, as I'm sure everyone has done before in their lives. The fact that those cells have been damaged means that they have to be replaced, and the way that your body replaces them is by copying existing cells that are already there. So this gets back to one of the tenets of cell theory that we previously discussed in this class, which is that all cells come from pre-existing cells, meaning that every single cell that is on the planet right now arose by being made a copy of a previous existing cell. Now, if I were to ask you guys what you think the most important part of a cell is to copy when going through this reproduction process, one thing that you might identify as being extremely important to copy and pass down to offspring cells is the cell's DNA. Given that the DNA is the blueprint for all of the activities that go on in the cell, you can imagine that this would be an important thing to copy. And so, indeed, before the cell replicates itself, DNA must be copied. That has to come first. And in copying its DNA, the cell can ensure that both of its daughter cells, which is the technical term for the two cells that emerge from this division process, will contain a full and complete set of DNA with all of the instructions that they need in order to live a healthy life. So with that in mind, before we move forward in the lecture, we need to both review and elaborate on what we know about DNA, which is a type of nucleic acid. We learned previously that nucleic acids are one of the 4 macromolecule classes, along with proteins, lipids, and carbohydrates. And we learned that they are made of smaller building blocks called nucleotides. Within those nucleotides, there are 3 different components. There is a sugar, a phosphate group, and a nitrogenous base. And that's where we left the discussion back in our first unit when we talked about nucleic acids. But here we need to go a little bit further, because these guys are the building blocks of DNA, and we need to know what DNA is really made of in order to have this discussion about how it is replicated and its significance to cellular reproduction. So it turns out that there are actually 4 different building blocks that make up DNA molecules. And within those building blocks, two of the components of the nucleotide are always the same. The phosphate group is always identical. You can see that across the four. The sugar down here is always identical. It is a sugar called deoxyribose, which is actually the namesake of DNA. Deoxyribonucleic acid stands for DNA, and that is why it's called DNA in the first place, because this sugar is called deoxyribose. And then the third component up here, the nitrogenous base, this third component can actually change, and it can be four different things. Those four different things are guanine, cytosine, adenine, and thymine. You can see that they have different chemical structures if you look at the highlighted portion of each nucleotide here. Scientists designate these usually with just their first letter abbreviation, so we call these the G's, the C's, the A's, and the T's. Another thing that it's important to know about DNA moving forward is that DNA is a double-stranded molecule, which means that when you have a string of nucleotides that are joined together end on end, as you have displayed here on the slide, every string like this is actually paired with a second string. And the two strings are held together by hydrogen bonding, a concept that we also learned about in our first unit, between the nitrogenous bases. And this occurs in a specific pattern, where the G nitrogenous bases are always attracted to the C nitrogenous bases and vice versa, and the A nitrogenous bases are always attracted to the T nitrogenous bases and vice versa. The complementary strands of DNA, which are called complementary because the sequence of letters on one is a complement to the sequence of letters on the other, they don't actually sit nice and flat like this. They actually coil up into this shape called a double helix, which is more of what you typically see represented in popular media for what DNA looks like. This is sort of a useful way for displaying it for the purposes of our class because we can see all the individual components, but this is what it really looks like when it's existing inside of living organisms. The significance of nucleic acids and DNA in particular is that the order of the letters, the A's, T's, G's, and C's, is like a four-letter language that spells out all of the instructions for making cellular processes happen. So this blueprint is written in a language that consists of four letters, A, T, G, and C, and that is how the cell learns what it should do, what it should make, how it should carry out its business. This leads us into another concept, which is the idea of a chromosome. DNA in living organisms is organized into structures that are called chromosomes. And a chromosome is a single, very long strand of DNA. On the previous slide, I presented you with an analogy, which is that the nitrogenous bases of A's, T's, G's, and C's are like the letters in the language of the cell. And so if the nitrogenous bases in the nucleotides are like the letters, then the chromosomes are like the books, a series of letters that are strung together in a particular order, all with a beginning and an end. The number and type of chromosomes that are found in living organisms can vary depending upon what the organism's identity is. Specifically, the chromosome number and structure differs between the two major classifications of cells which we discussed back in Chapter 4, those being prokaryotic cells and eukaryotic cells. So in this first review checkpoint here, I want you to describe to me two ways in which prokaryotic and eukaryotic cells differ from each other. So let's talk about the difference between prokaryotic chromosomes and eukaryotic chromosomes because there are consequences for how the cells divide in how their chromosomes are structured. Prokaryotic cells have circular chromosomes that are located in their nucleoid region. And when we say circular chromosomes, we mean it's a long strand of DNA wherein one end meets the other, so it doesn't have distinct ends. Prokaryotes can have either a single copy of their circular chromosome or they can have multiple copies of it. And if they have multiple copies of it, we can think about this as being like owning multiple copies of the same book. In addition to their one large circular chromosome, prokaryotes also often contain smaller circular pieces of DNA that we can kind of think about as like bonus DNA that are called plasmids, which also are found in the nucleoid region. Now, prokaryotic cells are rather simple, and so there's not a whole lot that goes into their replication. Prokaryotes replicate through a process known as binary fission. And if you think about what the words binary and fission mean, it really just literally means splitting in two. So binary means two, and fission, which is the opposite of fusion, means to split. Binary fission means splitting in two, and indeed, this is how prokaryotes replicate. What they do is they first make a copy of their circular chromosome, then they send one copy to each end of the cell before the cell is allowed to undergo cytokinesis, which is the splitting of the cell into two identical daughter cells. So it's very simple, very straightforward. Copy the chromosome, split the chromosome across two sides of the cell, and then split the cell in half. Now, when it comes to eukaryotic chromosomes and eukaryotic cell division, the process is much more complex, and so that's what we're going to spend the majority of the lecture talking about. Unlike prokaryotic chromosomes, eukaryotic chromosomes are made of a long strand of linear DNA rather than circular DNA, and that linear DNA is coiled around these little spherical proteins called histones. So here we see the DNA, here we see it coiling around these bunches of histones, and then those bunches actually form these supercoils, which gather together into the shape of the chromosome, which is what you see back here. And of course, all of these chromosomes are located inside of the nucleus of the cell because we're talking about a eukaryotic cell here, not a prokaryotic cell. Now, the depiction of the chromosome that you see right here as this structure with 4 arms sprouting from a central point, this is what chromosomes look like for only part of a cell's life cycle. For the other part of its life cycle, the chromosomes look like this, where they only have 2 arms instead of 4. Within this structure of the chromosome, the central point is called the centromere, and the arms that are coming out of the centromere are referred to as the P arm and the Q arm. The P arm is the smaller of the 2 arms, and what P stands for is petite. So that helps you remember that this is the smaller arm. Q doesn't stand for anything, it's just the letter that comes after P in the alphabet, and so it was assigned to the larger of the 2 arms. Within eukaryotes, most organisms have 2 sets of chromosomes. They have 2 copies, one that came from each of their parents. And because of this, they are described as being diploid. Diploid is a term used to refer to cells or organisms that have this double set of chromosomes because they are the product of sexual reproduction and 2 parents contributing their genetic information to their offspring. The number of total chromosomes that they can have varies quite widely from organism to organism. For example, based upon what we know now about the world of eukaryotic organisms, there have been documented cases of eukaryotes that have only a single pair of chromosomes, meaning 1 chromosome from mom and 1 chromosome from dad. That would be this ant right here. There are also eukaryotic organisms that have been documented to have up to 720 pairs of chromosomes. So 720 that came from mom and 720 that came from dad. And that would be this species of fern that you see right here. So obviously then the level of sophistication in the organism does not necessarily completely correlate to the number of chromosomes that it has. Another term that we want to introduce at this point, and that we will elaborate much more on later, is the term haploid. Haploid is the counterpoint to diploid, because whereas diploid is the descriptor for organisms or cells that have two copies of each chromosome, haploid is the descriptor for cells that only have a single copy of each chromosome instead of a full pair. In this chapter, we're not going to talk about haploid cells, but we are going to talk about them in the next chapter, so just be aware of what that term means and that it is coming up in the future. So, as we just learned, the number of chromosomes found in eukaryotic species can range quite widely from 1 to 720 pairs, but humans in particular have 23 pairs of chromosomes, or in other words, 46 total chromosomes, 23 from mom and 23 from dad. This would be like owning 23 different books, but you have two copies of each distinct book. What you see on the screen here is sort of a library of those books called a karyotype. And a karyotype is a tool for displaying an individual organism's unique set of chromosomes all lined up with their pair so that you can see them all together at the same time. This karyotype tool can be useful for diagnosing chromosomal disorders. For example, if a person has an extra copy of a chromosome. It can also be useful for visualizing the basis for biological sex because while the first 22 pairs of chromosomes here are considered autosomes, the last one, this 23rd pair, is considered the sex chromosomes. And these chromosomes are part of what forms the basis of biological sex. This individual right here has the chromosome pairing X and Y for its sex chromosomes, which means that this individual is most likely biologically male. An X chromosome paired with a second X chromosome would result in biological female. So let's talk a little bit more about what these pairs of chromosomes do. Every pair of chromosomes contains instructions or recipes for different processes and structures. For example, you are right here looking at a cartoon depiction of human chromosome number 4, which everyone has two copies of, one that came from their mother and one that came from their father. Along this human chromosome number 4, there are sets of instructions or different recipes, including one set of instructions, a segment of DNA that contains instructions for making a specific protein called alpha-synuclein. This protein, alpha-synuclein, is manufactured specifically in your nervous system. It's made in your brain cells and in your nerve cells throughout your body. Mutations in this segment of DNA are part of the cause of Parkinson's disease, which is a neurodegenerative disease that results in loss of motor control and often tremors in the hands and extremities that you may or may not be familiar with. Another thing that I want you guys to know is how we name the location or the locus of segments of DNA on a chromosome. So, in particular, the locus of the alpha-synuclein protein instructions on this chromosome is designated 4q22.1. So what does that mean? Well, the locus has three different components to it. The first number is the chromosome number. And we were just talking about human chromosome number 4. So it makes sense that 4 is the first number here. Q, you may have already deduced, is the arm, either the small or the large arm, so this letter will either be P or Q. And then the third number is the position of the gene on that arm. So the arms have been divided up into individual segments, and this particular gene or segment of DNA is located at 22.1 position. Now, we were looking at one chromosome number 4, but as I said, everyone has two of these guys, one from your mom and one from your dad. And these pairs of chromosomes contain instructions for the same processes and are called homologous chromosomes. The prefix homo means the same, and so homologous chromosomes contain the same sets of instructions. For example, every person has two chromosome number 4s, one from mom and one from dad, which means that your body actually has two separate sets of instructions for making this alpha-synuclein protein, one on the first chromosome and one on the second chromosome. However, it is possible that the contents of the instructions that are found on your homologous chromosomes differs between them. For example, the chromosome number 4 that you got from your mom could have correct instructions for making an effective alpha-synuclein protein, whereas the chromosome number 4 that you got from your dad could have mutated instructions, which would encode a defective protein. And this would put a person at risk for Parkinson's disease. So now that we've covered chromosomes, we can get back to the task at hand, which is really a discussion of how eukaryotic cells replicate or copy themselves. This occurs over the course of a process called the cell cycle, which really describes everything that happens to a cell from the time it is quote-unquote born to the time when it replicates. And this overall cell cycle is divided into 5 separate stages. We're going to walk through those stages and talk about what happens in them. And then finally, at the very end, we will see how the cell actually replicates itself to make 2 new daughter cells. The first stage, or phase, in the cell cycle is called the G1 phase. The G1 phase is the phase that initiates right after a new cell is created. And it is during this time that cells grow and develop and start to prepare for their eventual task of replicating. After the G1 phase comes the S phase. It is during the S phase that cells replicate all of their DNA, making a second copy of it, as well as all of their organelles. The S in S phase actually stands for synthesis, and synthesis is a word that means to create. So the S phase, or the synthesis phase, is when all of the extra DNA and organelles are created for the future daughter cells that are going to receive them. It is during the S phase that the chromosomes that we saw, the 2-armed structures, turn into the 4-armed structures that we also saw earlier in the lecture. So during G1 phase, a cell's DNA looks like this, and after S phase, it will look like this. The cell will replicate each arm of the chromosome so that it is paired with a second arm. And within the overall chromosome structure, a sister chromatid refers to 1/2 of the chromosome. So a P arm and a Q arm put together, of which each chromosome has 2. Here's an entire human karyotype and what it might look like before and after S phase. So before S phase, we see all the chromosomes only have 2 arms associated with them. After S phase, now they have 4 arms because all of the DNA has been doubled. It's still just tied together at that same centromere center point. So let's work on a little chromosome math here. Domestic dogs actually have more chromosomes than humans do. They have 39 pairs of chromosomes. That being the case, how many total chromosomes do dogs have? And in this one, I want you to tell me, after S phase, how many total sister chromatids would be found in a domestic dog cell? So now that we've talked about S phase, we're moving on to the next phase of the cell cycle, which is the G2 phase. During the G2 phase, like in the G1 phase, the cell is going to be growing, and now it's preparing for division because it has already replicated all of its DNA and organelles during the S phase, so at this point it's really seriously moving towards the cell splitting. Now these first 3 phases together, G1, S, and G2, are collectively called interphase because these are all of just the preparatory phases that occur before the actual division event, and so they're clumped together under the same broader term of interphase. As you can see, the cell spends most of its life in interphase, and a much smaller proportion of its life is spent in what is called M phase, or the last two cycles, or stages of the cell cycle, which are mitosis and cytokinesis. So we're going to move on now to talking about M phase, the division part, by going into a process called mitosis, where the cell divides its copies of DNA into two identical nuclei, which is the plural term for nucleus. So mitosis is defined as the process by which the cell divides its replicated chromosomes into two identical sets in two newly made nuclei. There are 4 distinct phases over the course of which mitosis is carried out. After interphase, the cell moves into prophase, then metaphase, then anaphase, and then finally telophase and cytokinesis. And some years ago, I found this image of a tattoo that someone got of the stages of mitosis taking place on their arm. And since I've seen this, it has always been my philosophy that if a student wants to get a tattoo of the phases of mitosis or any other depiction of cell biology, then you are free to use that on the exam. I will let you do that if you want to really go that hard and get a tattoo of it. Okay, let's get into now what happens in the stages of mitosis. Just before mitosis takes place, at the very end of G2 in interphase, the cell now has a single intact nucleus which has 2 full copies of its DNA inside of it. However, the DNA is not wound into the structure of chromosomes because remember, the chromosomes are made up of massive amounts of DNA that are wound around those histone proteins and then shaped into that multi-armed structure. But it takes effort and energy to keep it in the shape of that structure. And so when it doesn't have to, the cell will allow that DNA to exist in its loose form, which is called chromatin. And it effectively just looks like one big bowl of spaghetti inside of the nucleus here. It's one big mess. It is only when the first stage of mitosis begins, that first stage being prophase, that the DNA begins to condense and wind into that form that we recognize as the chromosome. We can see this happening in this image right here inside of the nucleus, where these little 4-armed structures are gradually taking shape. In addition, during prophase, the nuclear envelope, which is the membrane that surrounds the nucleus of the cell, breaks down. Centrioles, which are structures that are responsible for building another structure called spindle fibers, assemble these fibers and begin to stretch them across the cell. The spindle fibers are going to become important later, but in this depiction where you can see that happening is right here. The little orange things are the centrioles and the little blue things are the spindle fibers that are being constructed. Once those spindle fibers get big enough, then the newly formed chromosomes will attach themselves to the spindle fibers at their kinetochore, which is part of their centromere, that central structure. Here's an animation quickly depicting these different things happening in prophase. We can see at the beginning the condensation of the chromatin into the chromosome shape. And we can see the dissolving of the nuclear membrane, which releases the chromosomes into their environment. Here's another animation that is showing this happening in a real cell under the microscope. Although it's much harder to see this happening, you can get some sense of how the chromosomes are forming from the sort of chromatin big spaghetti mess in the nucleus. After prophase, the cell moves into a stage called metaphase. Metaphase is pretty simple. A lot happened in prophase. We saw the chromosomes form, the nuclear membrane break down, The spindle fibers form and stretch out across the cell, and the chromosomes attach themselves to those spindle fibers. Metaphase is relatively pretty simple because all that happens during metaphase is the chromosomes, which are now attached to the spindle fibers, will line themselves up at the equator of the cell. The equator is described as the midline of the cell. And just like the equator of the Earth is the line that we draw around the midline of the Earth, the equator of the cell is the same thing. And this is where the chromosomes line up during metaphase. Here's an animation depicting this happening. And again, here is a depiction of this happening in that same cell under the microscope. Now the next phase, the third phase, is called anaphase. And it is during anaphase that the kinetochore, the component of the centromere, which is attaching those chromosomes to the spindle fibers, that partially breaks down and allows the two halves of the chromosome to separate. These two halves, the sister chromatids, are then pulled by the tension of the spindle fibers to opposite ends of the cell. Here again we see an animation of this happening, with the sister chromatids separating and being pulled apart. And finally, we can see this happening again in the cell under the microscope. And this brings us to the fourth and final phase of mitosis, which is called telophase. So at the end of anaphase, the result should be that there are two identical sets of chromosomes clustered at either end of the cell because the sister chromatids are identical to each other and one was dragged to each side of the cell. Once they are in their proper location, the sister chromatids begin to decondense back into their loose and stringy form of chromatin, and a new nuclear envelope forms around each of the clusters of DNA. Also at this point, the spindle fibers are disassembled because they are no longer needed, and by the end of telophase, there are two identical nuclei contained within a single cell, which was the goal of mitosis. Here's a look now at the animation of telophase. And then finally, telophase happening in the living cell under the microscope that we've been tracking. Now, in this image right here under the microscope, we can actually see that the end of telophase starts to bleed into the actual splitting of the cell, which, as we talked about before, is called cytokinesis. Cytokinesis follows immediately after telophase, the last stage of mitosis, and it involves the cell splitting into two complete new daughter cells, which each have one complete nucleus and a full set of organelles. This cytokinesis process happens a little bit differently in animal cells versus plant cells because plant cells have a cell wall, which animal cells do not, which makes the process different between them. In animal cells, which you can see depicted in the cartoon at the top, a contractile ring is assembled from filaments of the cytoskeleton, and then that constricts around the middle of the cell, causing it to pinch in two. In plant cells, vesicles that come from the Golgi apparatus carry cell wall material to the equator of the cell, and then those vesicles fuse allowing their cell wall material to form what's called a cell plate, or a layer of cell wall that goes on to separate the two new daughter plant cells. So I showed you some little animations of each stage as we talked about them, but now I'm going to walk you through another microscopy video of mitosis happening in real time here. So at this point, you've seen both animations and actual videos of cells undergoing mitosis, and I'm going to present you with just a few pictures of cells that are still images captured at a particular stage of mitosis. In this image right here, I want you to describe what is happening in this cell and tell me what phase of mitosis it depicts. Likewise, in this one, tell me what is happening in this cell and also what phase of mitosis it depicts. And finally, go ahead and tell me what would be the result if a cell did not complete its tasks in S phase properly. Now, this checkpoint here is not actually a hypothetical question at all because things do go wrong during the cell cycle and mistakes are made. And this leads us into our next topic, which is a discussion of how the cell regulates its own progress through the cycle in order to make sure that it catches any mistakes that happen. There are 3 points throughout the cell cycle wherein the cell will stop, pause, and assess to see if everything is proceeding properly. These 3 different points are called checkpoints, and the checkpoints ensure that if something has gone wrong, then the cell can either correct it or stop the division process entirely. The first checkpoint that occurs during the cell cycle is called the G1 checkpoint, and it occurs toward the end of G1 phase. The question that the cell needs to answer at this point is, are conditions favorable for cell division to proceed? At the end of G1, all the cell has done is grow and develop. It has not yet really committed to the division process, which is more or less what happens once it enters S phase and begins to replicate all of its DNA. So at the end of G1, the cell will check to see if it's ready to go through S phase by determining whether or not its DNA has been damaged in any way, whether or not it has grown to an adequate size to support S phase, whether there are adequate nutritional stores, and finally, growth factors present for it to undergo S phase. If the cell passes that checkpoint, then it proceeds into S phase and then into G2 phase. And at the end of G2 phase, there comes the G2 checkpoint. At this point, the cell needs to answer the question of, are conditions right to undergo mitosis? Because remember, G2 is at the very end of interphase right before mitosis takes place. In the G2 checkpoint, the cell will check to determine whether all chromosomes have been properly created during S phase. It will also check to see if there has been any DNA damage. And finally, it will determine if its cell size is adequate to undergo mitosis. The third and final checkpoint in the cell cycle is called the M checkpoint, and this actually occurs during mitosis at the end of metaphase, just before anaphase takes place. The cell needs to answer the question, are conditions right to proceed with anaphase? You may remember that anaphase is the phase when the sister chromatids get separated and pulled apart to opposite ends of the cell. And because this is an irreversible step for the cell, it needs to check to determine whether all of the chromosomes are properly attached to the spindle fibers so that when anaphase takes place, they will get pulled to one side or the other properly. Now, there are certain proteins that are involved in carrying out these checkpoints throughout the cell cycle. An example of one such protein that is very well understood in humans is called p53. p53 is involved specifically in the G1 checkpoint, and it is involved in checking for DNA damage. The p53 protein is responsible for halting the cell cycle if catastrophic damage is detected, and then it will recruit other proteins to try to fix that damage. Even more important, if the damage cannot be fixed, then p53 will help initiate apoptosis, which is defined as cellular suicide if the damage cannot be repaired. So p53 is essential to the execution of the G1 checkpoint and either getting the cell back on track or destroying the cell if the damage is too great to where the cell cannot recover. Another example of a protein that is involved in cell cycle checkpoints is called retinoblastoma protein. This one is also involved in the G1 checkpoint, but it is involved in checking for adequate cell size. Retinoblastoma protein will block progression to the S phase until the proper size has been reached. So as you can imagine, proteins like p53 and retinoblastoma are extremely important. This brings about the question, what happens if these proteins fail? All of these proteins are considered to be a class of proteins called tumor suppressor proteins. And that's because they control the cell from replicating under bad conditions. This means that problems with p53 or problems with retinoblastoma protein or any other tumor suppressor protein can result in cancer, which is defined as uncontrolled and abnormal cell growth. p53 and retinoblastoma are kind of like the regulators. They are the bouncers who keep bad cells from getting through the cell cycle. If there's something wrong with that p53 or that retinoblastoma protein, then that means that the cell cycle can continue despite the problems that are happening with the cells that are proceeding through it. And when that out of control division occurs, the result can be tumorous growth or cancer. So if we have this understanding of cancer, why haven't we cured it? The reason is because every case of cancer is different. There are literally thousands of genes involved in regulating the cell cycle and producing these tumor suppressor proteins. And every case of cancer is caused by a different and unique set of mutations, which results in the tumor growth. But the thing that they have in common is that all of them exhibit out of control cell division because the cell cycle is not being regulated properly with these checkpoints. For example, 50% of tumors where the genetics of the tumor has been analyzed exhibit mutations in the p53 protein, which shows just how essential that particular protein is to stopping out of control cell growth and cancer. Another tumor suppressor protein that we know about in humans is the BCL2 tumor suppressor. And the BCL2 tumor suppressor controls cell apoptosis. If a cell had a mutated version of this BCL2, how would that affect its cell cycle regulation? And don't just answer this by saying there would be problems. I want you to tell me specifically what would be those problems. I have a few more checkpoints here to finish off this lecture and review some concepts that we went over earlier in the lecture. In this one, I want you to label the figure at the left, A, B, C, D, E, and F. And just to be clear, what E is referring to is this whole arm here, whereas F is referring to this strand of material that makes up the arm. Next, I want you to tell me whether the chromosome as you see it here would exist before or after S phase, and how do you know? And lastly, lamin A is a signaling protein that is embedded in the cell membrane. The locus of the lamin A gene is 1q22. So tell me, on which human chromosome, arm, and position can you find the recipe for making this protein? And this is the final checkpoint of this lecture. I will see you guys in the next lecture.